A nerve impulse, also known as an action potential, is an electrical signal that travels along the length of a neuron. Its generation and conduction depend on the rapid flow of ions across the neuron's membrane, which temporarily reverses the electrical charge. Neurons have a resting potential, which is the electrical charge difference across the membrane when the neuron is not transmitting an impulse. In this resting state, the inside of the neuron is more negatively charged compared to the outside. This is primarily due to the active transport of ions by the sodium-potassium pump, which pumps out 3 sodium ions, Ni circumflex plus, for every 2 potassium ions, K circumflex plus, it pumps in, and the presence of negatively charged proteins inside the cell. The generation of an action potential is triggered by a stimulus, for example a neurotransmitter from another neuron or sensory input, that causes the membrane potential to change. This process unfolds in 3 main phases: depolarization, if the stimulus is strong enough to reach a threshold potential, typically around -55 MV, voltage-gated sodium channels open. This causes a rapid influx of positively charged Na circumflex plus ions into the cell. As a result, the inside of the neuron becomes momentarily positive relative to the outside, reversing the membrane potential. Repolarization. Almost immediately after depolarization, the voltage-gated sodium channels close and voltage-gated potassium channels open. This allows positively charged K circumflex plus ions to rush out of the cell. The outflow of K circumflex plus ions restores the negative charge inside the neuron, bringing the membrane potential back towards its resting state. Hyperpolarization. In some cases, the potassium channels remain open for a brief period after repolarization, causing the membrane potential to become even more negative than the resting potential. This is known as hyperpolarization. Following this, the sodium-potassium pump and ion leak channels work to restore the neuron to its original resting potential. Once an action potential is generated, it's conducted along the axon of the neuron as a self-propagating wave of depolarization. The action potential doesn't actually travel, rather it triggers a new action potential in the adjacent section of the axon. There are 2 main types of conduction. Continuous conduction. This occurs in unmyelinated axons. The action potential propagates as a continuous wave along the entire length of the axon. The depolarization of one section of the membrane triggers the depolarization of the next section, and so on. This method is relatively slow. Saltatory conduction. This occurs in myelinated axons. The myelin sheath acts as an electrical insulator, preventing the flow of ions across the axon's membrane. The action potential jumps from one node of Ranvier, a gap in the myelin sheath, to the next. At these nodes, voltage-gated ion channels are concentrated, and the action potential is regenerated. This jumping or saltatory movement significantly increases the speed of nerve impulse conduction, making it much faster than continuous conduction.